Hello, fellow foodies. This is Dr. Cassandra Quave, and I'm here with Foodie Pharmacology, the science podcast for the food curious. On this week's episode, I've got one of my favorite mass spectrometrists that I know. Her name is Dr. Nadja Cech, and she's a Patricia Sullivan Distinguished Professor of Chemistry at the University of North Carolina, Greensboro. Now, Nadja has a really fascinating background as a childhood growing up around medicinal plants and has just really shared this amazing interest in the natural world and the chemistry of nature. And so I'm so excited to have her on the show. Thanks so much for coming, Nadja.
So great to be here, Cassie. I know we are just kindred spirits in that way in our appreciation of plant chemistry in the natural world. And it's just a huge pleasure to get to talk with you and an honor to be here today.
It's great. Well, why don't we start with just tell us a little bit about yourself. Like, what was the thing that really turned you on to medicinal plants?
Well, I would have to say that medicinal plants turned me on to medicinal plants. I kind of spent my childhood around plants a lot. And I would say I'm a student of nature. I grew up, my parents decided to raise me sort of off grid, me and my sister and my brother in a farm in rural Oregon. And at that time, it was a different time in terms of how much supervision children were given, and we were kind of given free reign of the farm and allowed to go into the woods and explore and build fairy houses and build forts and climb trees and also to garden. And so I think I was often writing stories by the creek or sketching in a sketchbook and looking at plants up close, and also hearing people talk about using plants to treat disease. And that's something that fascinated me. And I really wanted to understand how that worked from an early age. And so I thought I would be a botanist, and I really appreciate botany. But in college, I got really excited about chemistry and realized that there was a connection between chemistry and plants and the medicinal effectiveness of plants.
That's great. And you actually, if I remember correctly, you had a really early start in college too, right? When did you finish college?
I was kind of unschooled by my parents for my early educational years, and then I went to community college when I was 14. So I had a nice community college start, which was really excellent, and I fell in love with the university environment and kind of never left since then. But strangely enough, I have my PhD, but I don't have a high school diploma because I went straight to college when I was 14. So, but no one ever asked for the high school diploma once you have the PhD, so it works out.
That works great. That's great. Well, one of those tools that I know that you got fascinated with was mass spectrometry when you were doing in college and going into your PhD. Can you tell us a little bit about, like, what is this tool and the mechanics of how it works, and how can we use this to understand nature better?
Yes, I don't get that question very often, Cassie. So I have to say, I'm like, my little nerd heart is bursting with joy at this moment to have an opportunity to talk about mass spectrometry. I will say that mass spectrometry is a tool that's been around for quite more than 100 years, that is used to essentially weigh molecules. So, each molecule has a unique composition of carbon and hydrogen and oxygen atoms, or most do, and other elements that are combined together to give it a very unique signature in terms of its weight. So you can identify a molecule if you can figure out how much it weighs, or at least that's a piece of the information we need to identify a molecule. So a lot of times, we're interested in an extract from a plant that might have some interesting effect. Maybe it kills bacteria. Maybe it helps to reduce pain when you consume it. And the question is, which molecule in there is responsible for that effect? And mass spectrometry is a tool that we use both to separate all the molecules that are in there from each other, and then to weigh them, which gives us insight into which ones are there.
That's great. And you touched on something that's one of the bigger challenges in natural products research, and that's the mixtures, right? Because plants don't produce just that single molecule, but there are many different molecules. So you have the challenge in separating them and also understanding which ones are present. So let's say we have this machine, this mass spectrometer, and you have a plant extract that maybe you've created by steeping it in alcohol, and you've prepared it for analysis by the machine. What does the machine actually do to get that information from the extract?
Well, somehow you have to go from that liquid extract into what we call the gas phase. So we have to get the molecules to be able to be to be able to move through a series of lenses within the mass spectrometer that help to separate them out depending on how much they weigh. So you can kind of imagine it almost like one type of mass spectrometer, a time of flight mass spectrometer, which is the one I like to use as an example. Essentially what you do is you have a tube that all the molecules are going to fly down. And you can imagine if you give all the molecules the same push at the beginning of the tube, that the heavy ones are not going to move as quickly because they're going to be big and slow. And then the light ones, if you give them the same push, are going to move really quickly. And so you can measure how long it takes for the molecule to move down that tube. And then that information is used to, essentially the instrument is calibrated to convert the information about how long it takes the molecule to fly down a tube to then connect that to what the mass of the molecule was, which then eventually helps us know what the molecule is. So what happens when we put the extract into the mass spectrometer is first it's converted over to a gas. And then it goes through a series of lenses, and you can imagine them like lenses that are guiding light, but actually these are lenses that are designed to guide ions. And then eventually they're going to go into that time of flight mass analyzer and move down and then strike a detector. And when they strike the detector, they cause electrons to be released, which we pick up. And so that allows us to measure how long it took for them to go down the tube, because we wait until we see a signal of electrons, and that tells you that an ion flew down the tube.
That's so cool. It's just really cool. I totally geek out around mass spec, and I love it. I love it as a tool to investigate medicinal plants. And I'm wondering, what are some of your favorite plants that you've investigated using mass spec? Or was there one that you started studying earlier on in your career?
Oh, that's a great question. Well, so I'll tell you that I actually started my career in mass spectrometry, not studying plants, but studying blood, which was, I was an undergraduate student at a forensic laboratory, and we were trying to figure out how to identify species in blood based on the molecules in the blood. And that actually got me really interested. It turns out that hemoglobin is different depending on the species of animal it comes from.
Oh, wow.
And so we were weighing hemoglobin molecules and using that to identify what species of animal the blood came from, and actually I used that in my undergraduate career to help solve a crime where someone was accused of killing bald eagles. And we developed this technique that allowed us to identify bald eagle blood. So that was like my first introduction. It was a very all-American introduction to
like CSI. I love that.
Yeah, it was very CSI. So that was really cool. And there was something about like that moment of sitting in front of a mass spectrometer and looking at the screen and seeing these patterns come up and realizing that those patterns were telling me this was actually bald eagle blood, which was really like, I feel like that was a transitional moment for me. And actually early on in my career as a faculty member, I started analyzing plant extracts. And I had a similar feeling when I would actually, like, I knew about the molecules in the plant extract. For example, spilanthes. If any of you have ever tasted spilanthes before, they call them buzz buttons. They make your tongue really numb, and they make you like salivate and drool if you consume them. And I remember the first plant I ever analyzed on my mass spectrometer was a spilanthes extract, and I saw the predicted molecular weights of the molecules in the spilanthes that make your mouth tingle. And I was just like smitten with this idea that you could actually see that with the instrument, and that kind of sucked me in. And so we've continued to analyze all different kinds of plants in what's been almost 20 years now for me working on those kinds of questions. Most recently, we've been working on kratom, which is a plant that's used for treating pain and studying the complex molecular makeup of kratom with mass spectrometry.
That's fascinating. Yeah, and kratom is a plant that's been in the news a lot. There's a lot of debate over its legality, and if I understand correctly, there's also debate around which species are actually in trade. So, and I know you just published a really nice paper on kratom. Can you tell us a bit about what you've discovered?
Yeah, thanks, Cassie. We just published a paper in Scientific Reports about kratom as part of a National Center for Complementary and Integrative Health funded project on kratom, and we've actually been collaborating with a group that's doing clinical studies of kratom to see whether it causes interactions when people consume pharmaceutical drugs at the same time that they consume kratom or other pharmaceutical substances. In fact, there's some concerns around drug interactions with kratom, especially because people use kratom to treat pain, but also anecdotally, it's supposed to be helpful for treating addiction to opioids. So we have a lot of people that are co-consuming opioids with kratom, and research is showing that the kratom constituents of the kratom plant actually alter the way opioids are metabolized, and so there seems to be a higher risk of overdose if you consume the two at the same time. So that's kind of how we got into this. But we also, on the one hand, we really want to help the public to realize the safety concerns of co-consuming kratom with opioids. On the other hand, we recognize that there is an opioid crisis in the U.S. and, there were 42,000 deaths in 2016 due to opioid overdose, and the majority of those are due to prescription opioids. So people are having over-the-counter drugs that are ending up causing overdose, maybe intentional or unintentional. And so, we're really interested in whether kratom could be a potential alternative for treating pain that might be less likely to lead to addiction. And it does seem like the pharmacology of how kratom works is very different than opioids. And that's one of the things we talk about in this paper, and also something that's been talked about in other research studies, is that it seems like the alkaloids in kratom that are responsible for its effects on pain do not operate in the same way that the opioids do, even though they bind to the opioid receptors.
Oh, interesting.
And so you see that there's less of a potential, especially for the major alkaloids that are in the plant. There's less of a potential to lead to addiction among people who consume kratom than there is with opioids, and also that seems to be less of a likelihood for overdose. So, of course, I'm not a clinician. I'm not recommending that anyone consume kratom, but I'm really fascinated from a scientific perspective in the possibility of developing alternative strategies for treating pain that are less likely to lead to the risks of addiction and overdose.
No, absolutely. It is, as you mentioned, it's a huge issue, and it's in a way it's ironic that opioids are originally from plants as well. So we're looking at one plant remedy that's been used inappropriately that's led to some bad health outcomes, and then looking to other plants for some possible solutions.
Yeah, exactly. Great. Well said.
Yeah. And so some of the other work that you've done, I'm thinking back through some of your really fascinating papers over the years. You've also worked in other areas. I know you've done a lot of work on infection. You've done a lot of work also on plants like Goldenseal. Can you tell us a bit about those areas of research?
Sure, absolutely. We are very interested, actually, in many ways, we overlap in interest with your lab, Cassie, in this, in finding alternative ways to treat infection that possibly don't lead to the development of resistance, which is such a problem with antibiotics, or simply to finding new treatments for infection that might help to treat drug-resistant infections that have become resistant to antibiotics due to either use or overuse of antibiotics. And Goldenseal is one plant that we've looked at a lot. It's the use of Goldenseal to treat infection dates back to the Iroquois and Cherokee. At least that's the earliest documented use. And it's used topically. And it's a plant that grows in North Carolina, but also along the East Coast in the hardwood forests. So we've been interested in understanding how it works. And partly our interest in Goldenseal is that early on we showed that it seemed like there was more than one constituent in the plant that were responsible for its activity. So a lot of times, there's sort of this idea that you can take a plant and distill it down to a single molecule and find that single molecule that could then become a pharmaceutical drug. But plants don't always work that way. And, there's a complex relationship between all the different organisms that are either coexisting mutually or in competition in a natural environment. And it seems like Goldenseal is one plant that produces an antimicrobial alkaloid called berberine that does kill bacteria, but it also makes these flavonoids that enhance the antimicrobial activity of the alkaloids, and they do that by sort of preventing the alkaloids from getting pumped out of bacterial cells. So one way that bacteria become resistant to toxins is that they produce these pumps that help protect them from whatever's coming in. And if you can block those pumps, then you can actually kill the bacteria more effectively. So plants have figured out how to both produce toxic compounds and sort of gum up the pumps that the bacteria use to protect themselves against those toxins. And therefore, what we see is that we actually have better potency in Goldenseal using a complex extract than we do using a single isolated alkaloid from the plant against bacteria.
Yeah, and this process is known as synergy in the field,
yes.
Right? And I think it's just so reflective of so much of traditional medicine is around holistic approaches to health. And in a way, even at the chemical level, plants tend to be more holistic because they're using these. You have perhaps your lead active compound, but then you might have several other helpers that enhance the activity or perhaps boost the immune system, or as you mentioned, block these resistance mechanisms.
Exactly. Yeah, and it's tough. It's a complicated thing to study. And that's why, linking back to mass spectrometry, it's really cool to have these tools that allow us to comprehensively characterize all the different molecules that are present in a plant and start to be able to then connect those to the biological effects that we see, as opposed to assuming that it's a simple one-to-one correspondence.
Yeah. Well, and I think what's so cool about the technology that we have access to today is that we're basically learning how to read the language of nature, right? Is like
well said. Absolutely.
Yeah. You have people think of plants, and we're talking a lot about medicinal plants on this episode, but this is certainly relevant to many herbal medicines that are taken as foods or as dietary supplements or as teas. Or even some of our crops. You have plants may be sessile, but of course they're in constant communication with fungi and bacteria and
insects.
Yeah, exactly. It's like all of these, there's a lot going on when you look closer, and that just makes me think back to your childhood when you were there sketching and observing nature. I was very much, in the same boat on the other side of the country in South Florida. I was in a tree looking at, epiphytes and in the mud, in the swamp, and just had these. I wasn't a very good artist, but I had some pretty, wild journals of just that love for nature. I think that's something that's so important to instill in children and even among our graduate students.
Oh, yes.
Yeah. And I wanted to shift to talk a bit about this because I know that student training and mentorship are things that you hold near and dear to your heart. And these are things that you also promote around equity and diversity and how do we train the next generation of scientists. So I was wondering if you could tell me a little bit about those initiatives.
Absolutely. Yeah, in some ways, I think that what I have always thought about is trying to bring as many people to the table as possible when we're doing science, and recognizing that that's not just something that we do because it benefits those who have historically been marginalized, but because it benefits all of us. That having a team of people helps us be more creative, more innovative, better at solving problems, and also just learning about the world and stretching and growing and getting to explore new things and new foods and new cultures and new languages. And so, being in a scientific research group is really an excellent opportunity for that. And we also recognize that it's not always been the case in science that everyone is welcome, even though philosophically many of us ascribe to that idea. So I think there's a real need to acknowledge, how is the system set up to favor certain individuals, certain demographics, privilege certain people, and how can we try to address some of the barriers that are put in place for others, and what does that look like?
Yeah, I'm listening to an audiobook right now. I'm a voracious reader, as I know you are as well, and I'm listening to A Lab of One's Own by Rita Caldwell, and she's a scientist that really lived through this coming of age of becoming a scientist in a time when women weren't supposed to have labs of their own, and this is still something that's, we still have issues with equity in among different genders in the sciences, but it goes well beyond that, and I think the fight's still not over. Yeah.
Well, and I think you and I benefit in many ways from those women who came before
Absolutely.
us and became scientists at a time when women weren't supposed to have labs of their own. And so for us, I don't know about for you, Cassie, but for me, it was almost like it never really occurred to me not to be a scientist because I'm a woman. It wasn't really a conversation that was being had at the time that I was going through college. It was certainly accepted that I would be welcomed, but that's thanks to the people who came before me. And while I agree that there's still challenges, and I think I find that those challenges are greater now that I am a senior, distinguished professor. So I find myself more often in spaces that are primarily dominated by men than was the case when I was more junior or when I was a student. So there are challenges still, but those challenges for me are much smaller than the challenges, for example, of an African American or, indigenous people or people who've just been denied resources to start out with, and also those who visibly just look different than the status quo, or who are culturally really in a different space. So, the question becomes then, how do we turn around and afford that same opportunity to those people that was afforded to us as women in science? And I think women are really poised to do that because we are particularly sensitive to the historical discrimination that has happened, and we're eager to help create a more inclusive environment.
Absolutely. And I'm really heartened to see all the initiatives that are happening also in different scientific societies. We're both members of the American Society of Pharmacognosy, and it's really exciting that you were actually on a panel recently on the topics of diversity and inclusion. And I know that they just announced a new fellowship for students. Tell us a little bit about that.
Yeah, the American Society of Pharmacognosy just rolled out a summer fellowship for individuals from underrepresented groups in science, to support undergraduate researchers and to give them summer research stipends, full summer research support. We also, as a society, have just made the opportunity for people from minority serving institutions to have free membership. We're doing various workshops and webinars. We have an ambassadors program sending people out to give talks at minority serving institutions to sort of get other people, teach others about the field of natural products and give them the opportunity to participate. So, there's a lot that needs to be done. It's almost bottomless in a way, but it's really exciting to see people energized and trying to affect change, and to see that I have great optimism about the younger generation coming into science right now and their capacity to do things in a different way. It's really exciting to see. So I have tremendous optimism for sure.
That's great. Well, moving forward, what's next on your horizon? What kind of exciting things do you have cooking that you can share, both in your science and your other outreach activities?
Great question. Yeah, so it's been an interesting year for everyone, as you know. So, it's been a learning experience for a lot of us in terms of finding ways to stay connected in the midst of a pandemic, when a lot of our interactions are happening now virtually, and we're not seeing each other as much as we were. I'm really proud of my research group for continuing to do research in the midst of a pandemic and going into the lab and practicing, physical distancing from each other when it's hard to do, and wearing masks and working at odd hours of the day and all that. And and training each other over Zoom and making videos and just so creative. And they're really inspiring to me. And one of the ways that I have been connecting with them is to garden with them. I know you've been gardening too over the summer, Cassie.
Yes, I love it.
Yeah, I do too. So I've always been a gardener. But this summer was definitely a summer like no other in terms of not traveling. So we were able to garden more, and I manage a community garden in my neighborhood. And that's actually a source of some of the plant material we study in my research lab. And for a long time, I've brought students into the garden to sort of help them connect with sort of, if you will, the roots of their research. And we publish papers on plants that we harvest and grow. And so that's pretty cool to have students be able to go all the way from working on a plant, planting it, seeing it grow, harvesting it, taking in the lab, looking at what molecules are there. And I'm really excited to do even more of that. I think it ties into what we were just talking about in terms of increasing participation in STEM, that not everybody finds the lab by the same pathway. And one pathway that really excites students is to see this stuff happening see it in action. And so and some people have different skill sets, right? Like some people are really good at math when they come to college, but they might know nothing about how to plant a seed. And there's some people that might know how to grow plants, but have never really solved a differential equation. So, it's sort of a way to diversify what we're doing. So I've got a project now where I'm working with our campus to try to create some outdoor learning spaces that include a botanical research garden and also a food production garden. And I really want to tie that to the research we're doing and also to education. So that's a big project on the horizon right now that I've been putting a lot of energy into. We'll see what comes of that.
That sounds amazing. You've got to write this up as a how-to manual so other universities can replicate it. No, because I agree. There's just something about seeing the full life cycle and of having your hands in the earth and understanding, because really you're not going to understand these, as we mentioned, the chemical language of life if you don't take time to look at life, right?
Yeah.
It's
if all you see is some dead goo in a test tube, it doesn't tell you much about life. Yeah,
exactly.
It's true. Yeah, absolutely. I'll mention one more thing, Cassie. In terms of future projects, our lab just got funded in a collaborative project with a couple other universities that's designed at essentially developing new tools with mass spectrometry for studying the complexity of botanical natural products. So it's called the HiFan Center, the Center for High Content Functional Annotation of Natural Products, and it includes scientists at Simon Fraser University and the University of California, Santa Cruz. So there's sort of three universities that are all co-PIs on the project, co-directors of the project, if you will. And we're really trying to make the tools that are used, including mass spectrometry, for characterizing the complexity of natural products of plant medicines more accessible to the community and more effective to solve problems that are really unique to the natural products field. So I'm really excited to be a part of that project, and we're just embarking on 5 years of that. So we'll see where that goes. But that's kind of the nerd science piece, separate from the gardening and education.
That's a cool science piece. I mean, it's all cool science, but I'm so excited about the direction that projects like that are going, because most people, again, when you see the TV version of science, you have scientists in lab coat with their goggles and they put something into a machine and voila, they have the answer. And no, we have in a single leaf, you have hundreds, if not thousands of compounds, and we don't know what most of those things are. No. And so building out those tools is so critical.
We don't even know how many there are. Like you just said, hundreds, if not thousands. Nobody can even agree on the answer. So that's actually one of the questions we're working on right now is what is the diversity of chemistry in a single leaf, and how many molecules are there and which ones are there? And it's a tougher question to answer than one would think, considering how many years we've devoted to the study of plants. But I think we're going to get there.
Yeah. Well, and it's funny if you even think a little bit further back into the chemistry of plants, we focus a lot on what are called secondary metabolites. And that this was distinguish them from primary metabolites, which scientists study because they're important for the basic growth of the plant. And then there were all these other things. They're like these secondary things. Like, what are these for? And we're just starting to understand that like secondary metabolites are actually some of the most important metabolites to human health. Yeah.
Exactly. They are secondary, but not secondary.
Exactly. Cool. Well, I know one other thing I want to touch on before we wrap up is you also have a podcast of your own. Can you tell us a bit about it and where we can find it?
Absolutely. So I am a co-host of the podcast Yes and Cafe. And I would love to have anyone listen who would like to. And I think if you just Google Yes and Cafe at the University of North Carolina, Greensboro, you will find it. It is available through Podbean, and you can stream it for free from the internet as well. We talk with all different guests of stories of ordinary people who do extraordinary things.
Oh, that sounds fascinating and a great place to leave this because you are doing some really extraordinary things. And I would not call you ordinary.
I feel pretty ordinary, Cassie.
Well, thanks so much for coming on the show, Nadja. This was a lot of fun.
Absolutely.
You've been listening to Foodie Pharmacology, the science podcast for the food curious, recorded remotely on Skype during the COVID-19 isolation period. You can find all of our past episodes on our website. It's foodiepharmacology.com. You can also find us on iTunes, Podbean, and all other major streaming services. If you'd like to catch the video version of this interview, definitely check out our YouTube channel at Teach Ethnobotany and look for the Foodie Pharmacology playlist. Thanks so much for listening. Stay healthy out there, and I'll see you next time.